Slight acknowledgement done. I'm going to get onto something which leads into why I do this. This is a thing called a chiringa, and chiringas are a feature of the Central Australian peoples. You can think of it essentially, if you want to think of it in modern terms, as a stone key in a key value type of human database. And that is encoded in song and ritual, none of which, of course, I will supply despite the request for you, song and ritual not being my great thing. But the idea of each of these points, they represent a point that you can hang knowledge off. And it's actually by performing knowledge you get to keep it and to hold it. Okay, and these guys and these things were actually quite subtle ways of encoding most of the things of a culture. They're not got the knowledge in it themselves, they just provide the memory points and the waypoints, if you like, for that knowledge. And what I'm doing now I see is sort of kind of similar. Okay, I earn my living from teaching. You can tell it's the physical sciences from the wearable graph paper that we all wear as uniform. Okay. However, the place that I learnt was at Murdoch. It was a temple of learning. It was a great place pre-Hex then, and it was a fusion site of ideas. If there was a place that had your sorts of culture, it was pre-Hex Murdoch. Sadly, all of that has been annihilated by the cultural changes of the last 20 years. And the line that I followed, the path that I followed was this studying of these lampreys, and that is where most of my skill sets got hung onto, just like the Tringa, like the points on there. Old knowledge I find comfortable, like old friends. That's why I DIYBIO. Most of the time I spend talking to some bunch of kids about something relatively trivial but very, very worthwhile, and that has to be done to a high level. But old knowledge is like going back over my old knowledge and figuring out how I can do it at home is like being with old friends. It's a kind of comfort. Unperformed knowledge also becomes ghost knowledge. And this is one of the reasons and drivers for citizen science out there. My DIYBIO stuff is like a performance space. I am actually going over what I did before, reinventing, reworking to current and contemporary things, but it's actually keeping what I have and what I really liked, my old friends, if you like, alive. Okay, and that's how I recall, re-encode and re-embody my knowledge by doing it. Philosophical thought for you. Okay, it's an old joke. If a man speaks in a forest and his wife isn't there to hear him, and she's not, okay, is he still wrong? Okay, but the point behind it goes with that, doesn't the same thing happen to knowledge? The question is, has an almost non-state to it. Knowledge that actually isn't being worked over, embodied, performed and dealt with is exactly the same. It might be true in itself, but it's disconnected, it's a nothing. It's a void. Okay, so these are the reasons why I like to do DIYBIO. Just a couple of things to knock over. Firstly, knowledge and objects associate. You don't have knowledge without objects, without material things, without artifacts. Even Pythagoras is associated with a triangle, and you can't have anything much more abstract than that. And knowledge is always traded, it isn't free. I had a Hex debt, admittedly, it was from my teaching, not from my PhD, to prove it. Knowledge is always traded, and it always has been, and it has always been bought. Think of the last time you went to see a doctor. It has been this way for all of our time. And knowledge, I'd say, can be mined like Bitcoin from nature. We call that research. Okay, like Bitcoin, objects, time, and money are required. So objects form a central bit of the gathering of knowledge. And the other thing is, again, coming back to emphasize at the end of that, unperformed knowledge is lost. Okay, so why op shops? Okay, why op shops? These are some of the ones that I use, and these are their, if you like, visual identities, their logos. Okay, and the art of the op shop, if you like, becomes its embodiment, that you recognise, that you can see. Art and the aesthetic is very important to do so. These are the ones I use. Okay, why op shops? They're cheap. Yeah, I'm Dr. Cheap. I'm known by my students as Dr. Cheap. Okay? They're also well made, because by the time that it's got to the op shop, the crap is in the landfill. The good stuff is actually there, and it's gone. They also represent, they also have older technologies which aren't so tightly integrated. Those things, you can't do very much with them, but you can take an old camera apart and you can get a high voltage supply out of it, which is how I've made things like Geiger counters. And one of the ways in which I'll make a high voltage, how I have made a high voltage capillary electrophoresis setup. That's something else that I will publish a little later over the holidays. It's sitting in a box waiting to be done. There's a bigger and different range of things, and if you like, it's no tragedy if it doesn't survive. Okay, it was going in the bin anyway. Okay. And my philosophy of that is you can think of an op shop as a playground of mechanical puns. Okay, sleights of hands, possible uses, other things like that. Who drinks coffee? Who drinks pod coffee? You know, nice. I like pod coffee. I bat out of helling it at 6:45 to get to the train. I'll chuck in a pod. Oh yes, down it goes. Won't even touch the side. Can you think anything useful to do with the pods? Come up with something amongst yourself. What would you do with a coffee pod? What can you do? Have you ever looked inside one?
The grounds or the actual plastic pod?
Oh, all of it. The grounds are great. You chuck them on the things you want to keep the snails off, because caffeine is quite a good molluscicide, incidentally, and it's quite reasonable. But there's an actual nice little fritted mesh and a few other things. Okay. Any gardeners? You're welcome. I have 6 of these. You may have one. That's what I do with my coffee pots. Okay. A mechanical pump. An example. You can have that. That's a sundew, a South African Drosophila capensis, if you want it. Stick yeah, they are.
Yeah, they're
carnivorous. Yeah.
I'll
leave there. You can find them. No. A sort of gnat. But at any rate, the point that I am making to you is this idea of mechanical puns. Here's some more of them. Okay, low speed, high capacity protoplast centrifuge, if you're trying to make that. You'll notice a salad spinner. Okay, they cost about $1.50 at the op shops. As I said, I'm not renowned for my extravagance in this particular thing, certainly not in those things. Okay, you want fridge reagent stores? If you're DIYBIO, this is the place to go. You got an ice bill, costs about $4 or $5 a time. At 50 cents a piece, you can sort of set up an ice rack that will run a DIYBIO lab for pretty much ever. If you just have the protocol of filling it up, putting it in after use, then everyone can use. It's always available and it's always convenient. These guys would easily give me the lot for $10 if I were nasty, and I tend not to be with op shops. 5, I reckon. Okay. Ice bath or steam on one of those inside the other. They're quite good for keeping things cold. Outside with ice, inside with whatever liquid or homogeneous thing you want homogenized or something like that. They're quite good for heating and cooling systems and control of that. Of interest to you people, and it's one of the things that I think I might go with, is the possibility of all the old printers that are sitting along the roadside. These are the cartridges for them. They go at 50 cents a piece. 50 cents, $1, 50 cents, these ones. Okay, so clean out the ink, get a couple, red, green, blue, something you want to grow with some alginate in it, something you want to grow with just some calcium as a setter, and then, you know, say maybe 2 or 3 different cell types. You're quite possible to make a little mosaic of things. Okay, projects. First one of the ones that got me into was only a hybrid shot was gas chromatography, and this is one of the earlier things that I made. This is a gas chromatograph. The detector that I use is actually a flame detector for halogenated solvents, and it's more like a proof of concept than anything else. It does work. I can actually resolve things quite nicely with that. I can also, with a little Arduino here, I can program the jacketed column. You see there's a borosilicate glass, bit of glass going through the end of that with some silica gel in it. I can program that to for different temperatures. And the sorts of things that I was able to, from the lab notebook, which is one of those exercise book things. Relatively simple circuit. Don't want to blind you with bullshit with it, baffle you with bullshit with it, I mean, but you can sort of put together one of those things quite easy, and all of these commodity type of items. Okay, that's what it looks like. So you get no bleed when you run it at room temperature, but you can sort of sweat it off at 55 degrees, and you can, by putting a program on the temperature of the column, like you can run it like any other glass chromatograph. The sensitivity wasn't high. The detector was a 20 cent. Did I mention that I was cheap? Actually, it wasn't 20 cents. I took it out of one of the cameras that I had stashed it away. Photocell, so it's not. It's a cadmium sulfide thing, not renowned for their sensitivity. And I just basically banged it up on a hook, photocell on this side, flame on that side, gas output, and produced it. But I could, quite easily. Discriminate. You can see the columns overloaded by the tailing and all the rest of it, but hey, proof of concept, get it out there, published through to one of the amateur science groups, the Science Madness, which are a generalised international loony bin of chemistry nerds, just like me. So, and yeah, very, very very useful thing to do. And that's the other thing with performance. You do it and then you sort of put it out and share. So what I'm keeping alive is a large amount of time that I spent, in this. I've now had to move away from that. Mortgage, kid, school fees, etc., wearable graph paper. Okay, is this October? It must be time for Markovnikov's rule again, like that. But this is actually how I keep it alive, but of course the performance part of it is important. Maybe someone else can get something out of it too. Okay, Geiger tube. This is made from a 50-cent old film camera and a piece of PVC pipe. Not new PVC pipe, you'll notice from the yellow that it is from an old retic project I had. Did I mention I was cheap? Okay, right, so yeah, there's that idea going through there with that. Basically Arduino, $20 Russian, the best quality of Russian tube that I get, SBM20. There are lots of them sold around from the old Soviet nuclear programs. The Arduinos R5, they're a little bit less. The LCD Shield is probably less now too, but I built this back in 2011, 2010, I think, and the remainder was junk box. Flywire was my own innovation. It just allows you to measure very weak particles. Some things that are supposed to be alpha emitters I can see on that. I reckon that may well be crap. I reckon I might be looking at the Auger electron rearrangements, which are very, very low energy once the nucleus has spat itself out. However, it's an alpha emitter, okay. Everyone else who makes anything like that says that, and it's good enough for me. All in the story. Right. This is a workshop that I'd worked up, actually. I sort of got it to a point where I could get some of my kids to do some things. This is a quantitative thin layer chromatography. So I ran a project for a Science Talent Quest project where I had 3 absolutely hard-working and dedicated kids who picked this up, and they were amazing. Like, they had the research ethos. They didn't fall over when something went wrong, and they spent every Wednesday, I think it was, working away at this after school for a while. So basically we take some talc powder, we mix it with some gypsum, we spread it over microscope slides, we dry it, we rub it on some paper, A4, because, you know, why would you buy 1500 grit paper when the titanium dioxide in this makes it about 1800 grit paper? You know, I think I might have mentioned my extravagance before. Okay, but it does a nice job and it cleans it off nicely. When you run it through a peak, and I don't have the actual development of it, you can see you get these. Let me show you. Series of peak area under the curve, or pixels that show up depending what you stain it with, and then you can use a cheap bit of software called NIH Image, when I was a youngster, but now called Image J because it's all been ported from C++ to Java. About 20 years ago actually, I am showing my age, and you can get yourself a nice standard curve. I can measure paracetamol I'll pick something like urine, water, whatever, using this technique, and a moderate sensitivity. I wouldn't write home and tell your mum about it. If I had an HPLC, that's what I'd go for, but I don't. So I'll do that. That's next year's project, by the way. We will. Okay, so we get on to plant tissue culture at Willerton. That was a Willerton project back there. This is one now. The attributes I'm looking for is it must be cheap, you know. It's not true, like things do attract, it must be cheap. It must be cleanable and reusable. So, you know, I'm not really that interested in the, polystyrene plates that are used in the lab, because basically I have difficulty getting them sterile. And if I autoclave them, they look really, 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 really tiny and crinkly. Okay, easy to make. And because I'm dealing with kids, no mains wiring or certified mains wiring. That's an absolute given. Now, I don't mind taking it apart myself. You wouldn't believe what I would be happy to take apart myself. I've got kids around this. If someone, you know, even gets hay fever, it might be because someone will make a very long bow, but it's because they're too close to something that wasn't done well electrical, and there's no mechanistic reason for it. But you know, courts don't don't rely on mechanisms. Okay, programmable day-night cycle. Low temperature and a suitable light spectrum. All of these are problems. Okay, you can see where this led. Not all puns are mechanical. Like the modern LED lights are superb for this, and they're what the aquarists are using. Okay, so light quality, $29 at JCar. I don't think you can even begin to realise how much it hurt me, that $29. It was $29. You know what you can buy in an op shop for $29? However, you know, I did succumb. It was the best way of doing it. And I checked out the spectra. What I was after was a fairly good continuous or semi-continuous spectrum over this region, over all the regions that the plants might photosynthesize in. Hey Harris, I thought you said you were cheap, you know, you got a spectrometer to do this sort of thing. Well, yeah, both are right. Spectral Workbench make a papercraft spectrometer. It's a nice little DIY sort of science project. You fold it up into a box, you put a bit of CD that's broken or thrown away on there as your diffraction gradient, and you photograph it with a camera. If you use one of these compact fluorescent lights, you can pull out the mercury vapor wavelength and some of the well-known fluors, and you can calibrate the thing. Probably, I reckon, maybe 10 nanometers, plus or minus 10 nanometers, but you know, beggars can't be choosers, and it does quite a nice job. At any rate.
Is that, sorry, is that that's not a rewritable CD? Or is it?
Oh, just a CD. The idea is something with lots and lots of holes in it. So rewritable would do, whatever you've got. So to go jump into the dumpster, grab one out, stick it in the microwave, go ooh ah as the metallic bits sheened off, and then you've got all the pits in it. It's really good. And you know, if you like what you saw again, maybe you need another one, so do it again, ooh ah, you know, and then, but you know, make sure you do it while your significant other is out of the house, because she labours under the illusion that it's her microwave, you know. Oh, you know. What can I say? Incubator parts, that's how simple it is to make a plant tissue culture incubator. This guy up here was an Esky, cost me $12 at Sammy's. It was solely because my mate wasn't there. I'd have got it for, I reckon, about or 5 or 6, because he always gives out stuff. But you also have to be there at the right time, and there was someone all very earnest and who was there, and yeah, okay, well, fine, that's fine. $12 as compared to 40, that's all right by me. One of these things come from Kmart. They're a programmable mechanical timer. Old technology, very old technology. You look at a kid like that and say it's programmable, and they'll raise their eyebrow at you. So what do you mean by programmable? Well, that's just complete BS, but it isn't. You can set your time to your day-night cycle. It works well. There's my LED light from JCar. It's the cheapest, are you not surprised, of their range of things there, and it meets my requirement of being fully sealed, encapsulated, and all the electrical work done by someone else. And in this way, that's how you can put together quite a good range of things. I use a thing called Google Science Journal for light intensity. I'm not that fond of Google Science Journal all the time, because there can be problems in getting your data out of it. It's like the old joke. Our backup and restore solution is free. Well, the backup part of it is, but our restore consultant is going to cost you most of your budget for the next year. Okay, well, it's a little bit like this as well. It goes in very easily and it looks very nice. However, the problem is that I've had some difficulty getting it out at the moment. I'm told it's because I registered as a student rather than as myself, and they don't allow exchange of data for under 13-year-olds, which I must have clicked something that I didn't know about at the time. Oh well, doesn't matter. It's a very capable light meter. I get about 11K. I went out to the Jarrah forest where the species that I was interested were growing, and I measured that I was also anything from about 4K through to about 16K in that, but mostly dappled sunlight would be met for that. U-Beauty, that's good enough for government work. We'll have that, thank you very much. Does the job nicely. Okay, so once I've got that out, you get it up and you put it out to Instructables. That's part of the culture of DIYBIO. You share. It is also, it's pleasant. It's a performance piece in a sense, if you like, but also it's got another meaning to it as well, that there are other people who want to run with it, and you might actually learn something, because there are people smarter than you out in the world who will see things that you don't see, and then you get to rip off their ideas, and then they become yours, and everyone thinks that you're cleverer more than you usually are, than you actually are. Okay, so out to Instructables. I thoroughly recommend Instructables as a place to go for it. Tissue culture techniques, okay, aka rip off a first-year plants physiology lab. That's literally what I did. Okay, why would you need to reinvent something that had already been reinvented before? Well, so be it. There are heaps of these things. Base it on the Pan-African violet. They're very boring. You probably can do better, but everyone grows them because they always work, and they're kind of exotic, unless you happen to sit on one of the world's best temperate biodiversity hotspots, and then of course they're just boring, but not everyone is as fortunate as we in their location. Okay, my comment is don't be beige. Go to somewhere unique like the Wildflower Society, and the plants that I gave to the kids we call organ donors. And basically, for $4 you buy a little tube like that, you give it to a kid, or a couple of kids, they hack a few bits off it, they think it's really great. Even tell them that it's surgery, perhaps they do enjoy that aspect of it. And especially if there's some criteria to it, like look for a nice meristem, take it out, clean it, handle it properly, don't drop it in the dirt, all the rest of it. That sort of technical detail of actually doing is really good for them. Okay, find a hobbyist-friendly supplier in Australia. You can get all of this through eBay. It's all advertised. It's of dubious legality, I think. what can and what can't be brought in, but you know, certainly anecdotally across the network, I know that you can, anything you want pretty much will come in from there. It's much nicer to have a relationship with a good Australian place. Austrotech are superb. They like hobbyists, they like independent people, and they will make it work for you. I have nothing but high praise for those guys, and they're an agent for the bigger tissue culture suppliers in the States. So, yep, support local is good, but also it just makes your life easier on a whole range of things. Okay, when stuff in the reject shop grows up, it wants to be in an op shop. And really, you know, this is what cheap new looks like, but you can do a whole load of stuff with it. I will just give you a brief squiz. When I say $20 meth in $2 hairspray, this guy here, okay? How do you get 70% meths? Well, you tip out 300 ml from 1 litre bottle, you put a bit of tape along that particular mark, and also before that the mark that was at the top, and you've got your standard meth bottle that you can fill up to 70%, fill up the rest with water, and basically you have lots and lots of running ethanol. Okay, ages ago, people figured that, you know, we're going to put methanol in methylated spirits because we don't want those evil peasants to rip us off by not paying tax. As we moved more towards the kind of, if you like, not welfare, but sort of integrated society that where things are bought on the public health, someone said, "That's crazy. You're taking our hardcore alcoholics and you're making them blind. That's going to put another $50,000 a piece on them before they die. What sort of royalties are you trying to save on this thing?" So now there's almost no methanol in Australian methylated spirits. It's very good for this purpose. There's a thing called denatonium in it that is a bittering agent, but not much more, and that works very well with tissue culture. Okay, I see. You meet my mark of approval, you guys do. One of the best culture systems around. Nice and cheap, recyclable, reusable, about $1. Okay, so this is what I call a not shot bottle. It's came from China, and they're about $3 or $4. This is one of Austrotech's many reused tissue culture things, and this is something I bought from eBay, I think about 2003, 2002, from an old guy who was retiring as a science teacher, and these are the glass Petri dishes. They're still around at the moment. All you need, what you do, spray the inside vigorously, you know, like don't be cheap on this one, cover it with ethanol. See how the slick on the bottom? That was dry when it started. Now you've got a sterile environment, you've got an airflow that you're not intruding to. Gloves covered with a spray of ethanol and a weight, and you can do pretty much all your tissue culture in the kitchen. I did. Okay, and then finally again the principle of this is to get it figured out, share to instructables. These are stylidia, trigger plants. Trigger plants are plants that bash animals, you know. As part of their sexual ritual, they bash animals. Here's the idea. Insect glands, these plants have a fused anther and stigma. They bang the side of the animal, transfer pollen. There are about 130 species in Western Australia, and hybrids are not good for organisms to do. They don't transfer their genes well. So, there are butt specialists, there are head specialists, there are wing specialists, and there's every other kind of specialist in between. The species pretty much divide up between themselves the part of the insect they hit. It's a beautiful biology question on sympatric speciation if you are interested in it. Okay, so here's my callus, put into my rooting media, and there's the guy growing at the moment, considering flowering at this particular time. Okay, and then once you've got it up, we of course unleash the kids. limited number of what I actually can release, limited to what I could find on that was still on my phone last night after I'd scrubbed it off. But basically the kids actually do enjoy quite a lot of the ritual of making up the material and preparing for a run of tissue culture and weighing out the chemicals here. And this was our tissue culture club that we run, and that's pretty much where I fit into things, and that's why I'm with, if you like, the DIYBIO kind of people. It is a performance after all, and that's how I remember and keep it alive for myself. Questions?
First of all, thank you.
Just quickly, sorry.
I was going to say no answer came the stern and forbidding reply, but yes, go on.
Lampreys. Have you ever tried lamprey?
No, I never ate it, but this, you've got to understand, I used to have tanks with up to about 300 of them, and they migratory creatures. When they migrate, occasionally into the tubing between tank to tank, they block the flow. I have smelt, not a huge number, because it didn't happen all that often, but I've smelt enough dead lampreys not to be attracted towards them.
Yeah, they're supposedly quite delicious.
They are. They were supposedly an aphrodisiac in the Middle Ages. Basically anything with a bit of iron, some good marine oils and high protein was an aphrodisiac. If you're that big, filled with intestinal parasites and from the Middle Ages, you know, it was a nutritional sort of thing, and that I don't think there was you know, anything further than that to it, other than being a really, really good and abundant feed. And of course, as they come up en masse, you know, you get a huge surge of calories, and you know, if you're just about starving and you're looking for time, or your physiology is looking time for reproduce, that's one of the best times to do so.
I enjoy some lamprey trivia.
Sorry?
I like lamprey trivia, so yeah, thank you.
Oh yes, yeah. I like all kinds of trivia, can't you tell? Yeah. Any more?
I've definitely used Ostrotec that we used to get the plant tissue culture stuff. So you can
We're running a plant tissue culture workshop in October with carrots. You know.
Oh, okay, yeah.
So I just like go down to Farmer Jack and grab a few carrots, like the smaller baby carrots, and chop them up in the lab and get some calluses out of them. So it's fun to see that process, and yours is far cheaper than ours.
Thank you.
It's,
you know,
it's impressive.
we'd have to fight if otherwise. You know, this is my reputation.
But then, with the actual box that you have, do you have a heap of it?